Hey there! This is a tutorial on how to do a Punnett square and some of the reasons behind why we do those. So I'm going to go ahead and get started. Before we get started, you need a piece of paper and you need to write down everything that's in yellow for these notes. So I'm going to go ahead and get started. Just to review before we jump into Punnetts, I need to review some terms. So homozygous or purebred, dominant would be the genotype, for example, big P, we're using P's. Heterozygous, which means different alleles, is going to be big P, little p. Another word for that's hybrid. Homozygous purebred recessive is little p. So I could use either of these words, homozygous or purebred, interchangeably. I can use either of these words, heterozygous or hybrid, also interchangeably. And I will use each of these words at different times and they mean the same thing, because hetero means different, so we have two different letters here. Homo means same, so we have the same letters here. Homozygous, same letters here. Dominant, capital letters, it means that alleles are going to show if they're present. Recessive means that in a situation like a heterozygous individual, this lowercase recessive allele will be masked or hidden. One more thing to review, which we may not have covered in class, but I want to make an emphasis of it before you start your Punnetts, is that you will make your letters different for credit. It's very difficult to tell most of these letters apart from each other because their capital and their lowercase look the same. So please make them different somehow. Here I have put a strike through, put a line through the lowercase ones. Sometimes I'll use cursive for the lowercase. Please keep the uppercase dominant alleles in print, and you can change the lower ones however you like. Just make sure they're the same letter. What letter should you use for a trait? I don't know if that's been covered either. You should use a letter of the dominant allele. So a good example would be if the trait is hair color, maybe for guinea pigs or some animal, and you have two different alleles, brown or red, and if brown is dominant to red, we would use a B for brown, B for the trait, and big B for brown and little b for red, because the brown allele is dominant for this hair color trait. So I think that's good review. Let's move on. A Punnett square, the purpose is to predict the possible offspring of a cross between two parents. So we will get started, and you may have to push pause to write some of this down, but I'm going to continue. The steps of a Punnett is to, number one, define the trait and alleles. I already reviewed those a little bit, but we are going to use for this example the trait being flower color. We're going to talk about Mendel's pea plants either being purple or white, where purple is dominant to white. So we use the P because it's a dominant allele, and we use big P for purple, and the little p is a recessive allele for white. We need next the genotypes of the two parents. Now this is where every problem is going to be different. So you might note that, okay? So in this example, I'm saying mom and dad, both parents, are heterozygous or hybrid for purple flowers in this example. And so their genotypes are, since they're heterozygous, hetero means different, they each have a big P and a little p. So here's one parent, and we're going to write an X showing that they're going to mate. So this is the cross, and here's the other parent. So both are heterozygous or hybrid for purple flowers. So that's how we write each parent. And in every genetics problem, this is the first thing I want to see. Besides maybe the trait defined and the alleles for each trait, the next thing I want to see are the parents' genotypes. Remember, genotypes are the genes they have. After that, you're going to write the possible alleles each parent can give in their gametes under their genotype. So here is the cross from the last page, and this parent can give a big P or a little p. Let's say this is the mom, so in her egg, and she only gives one for each egg. She can't give both of her chromosomes. She only gives one of them. Excuse me, and the father gives in his sperm either a big P or a little p because he has one of each. It won't always look like this, but in this cross it does. So if these parents are heterozygous for flower color, they can make gametes with only one allele, either a big P or a little p, and we list the possibilities of what they can give. Now we know only one egg and one sperm will match up. So why did I make eggs and sperm? Because we are predicting what's going to happen when they mate together, and we need one egg and one sperm. The law of segregation states that two alleles for a trait separate when gametes are formed. Now you already know this from our meiosis chapter. Gametes carry only one allele for each inherited trait. So what that means is that when we start out with, these are two parents that are like big R, little r. Let's just take the R's. We won't worry about the Y's. So big R, little r, it would be like big P, little p in our example. When they, this is metaphase 1 of meiosis, and when they line up here and then they're going to separate, the big R goes here in the first meiotic division, and the little r goes here, and then this is metaphase 2 as it says, and what's happening is that each of the alleles go in separate gametes. So let's read it again. The law of segregation, two alleles for a trait, like big R, little r, separate when gametes are formed. So you don't get a big R, little r in these gametes. They separate. There's a big R here, a big R here, a little r here, and a little r here. So this is like one parent forming gametes right here. Gametes carry only one allele for each inherited trait. So that's kind of what we're showing when we show, when we put the possible alleles with the egg and the sperm. The next step is to make a 4-square box, which is easy. Then, with what you've done here, you're going to take the one parent's possible alleles and list them in each row, and the other parent's possible alleles and list them in each column. And so what we're doing is we're saying if this parent and this parent mate, this is what could happen. Their offspring could, upon fertilization and making of a zygote, they could, this egg and this sperm mate, they would bring big P and a big P. And that's your next step, is to fill in the punnet. So if this parent, or if this egg and this sperm happen to make an offspring, it will be big P, big P. If this egg and this sperm make an offspring, it'll be big P, little p. If this egg and this sperm make an offspring, it'll also be big P, little p, but we're going to always put the dominant allele first if it's heterozygous. And so we don't put little p, big P. And then finally, if this egg and this sperm mate, sorry, didn't mean to click, then you will get little p, little p. So this is how to fill in the punnet, and this is what the outside of the punnet, what those actually stand for. So we have made a punnet, and we know how to do that. How do we use it? Because that's really the most important part. We look at a punnet to see what the ratios are of the genotypes and phenotypes of the offspring. So after we make this, the next thing to do is to write out the genotypic ratio. It's easy. All you have to do is look at the boxes, and these are the possible offspring of these parents. So you have 1 big P to 2 big P little p's to 1 little p. So that's the genotypic ratio. You put a colon between each for a ratio, 1 to 2 to 1. And then the phenotypic ratio is 3 purple to 1 white. So this would have, this offspring would be purple, this one would be purple, purple, but this one would be white. So we get 3 purple to 1 white, and we write it like that. Now, what can we do with these ratios? We're almost finished. We can actually predict offspring. We do a punnet so that we can actually predict what two parents will actually, probabilities of what their kids will look like. For example, if these parents, big P little p times big P little p, had 20 offspring, these are pea plants, so they could easily have 20 little baby peas, how many would be white? And so you use the number 20, and you look at your punnet, or your ratios, and you say, well, the ratio is 3 to 1. So that means 1/4, 1 out of 4, are white. Well, if 1 out of 4 are white, that's really 25%. Well, 1/4 of 20, which is how many we had total, is 5, because 5 times 4 is 20. So 5 offspring would actually be white. And so this is why, now it's not going to be exactly 5, but it probably would be close. One more term that we haven't been over, and I just want to throw it in, is that a monohybrid cross. So mono means one, so one trait. Hybrid means they're both parents are big P, little p. So a monohybrid cross is where both parents are hybrid for the trait. So what we just did in our example was a monohybrid cross. But that's not all of the one trait crosses you can do. So what other crosses are possible besides a monohybrid cross? Well, all of these are possible. So if they're both purebred dominant, then this is the cross. And then the Punnett would look different, as would all the ratios and probabilities. And then on down here, you can look at each of one of these as you like, but they're all different. So, and it doesn't really matter. I wouldn't switch these parents and say big P, little p times over here and big P over here. It doesn't matter which parent's on which side. But these are all actually different. There are none that are the same. So basically those are the possibilities of different one trait crosses. These are called one trait crosses. They're all one trait crosses. This is the only monohybrid cross because it's one trait, because you have only P's, and then they're both hybrid for the trait. Both parents are hybrid. So hopefully this is helpful. Do your practice worksheet, and we will answer some questions in class.